we come to the last talk before our break by Svenja Kaspers from Jülich. Svenja will speak about high-resolution fiber tract visualization in human occipital cortex.
Yeah, hello everyone, thank you very much. As you already heard a lot about cortical mapping and the cortical areas, and specifically in the last talk about retinotopy, my talk will focus about the other part, which is also important to understand function and functional anatomy, which will be the fiber tracts. And I hope I can convince you that also understanding the precise location and course of the fiber tract is important to understand the functional anatomy, particularly here in this case of the visual cortex. So, let's see if this, yeah, it's Mac, but it doesn't do any. No. Okay, thanks. So I will subdivide my talk into two parts. The first part will be about the projection and association fibers, so the ones which run into the visual cortex and which connect the visual areas with each other. And the second part will be about the transcallosal fibers, which are very important, as you heard this morning, for binocular vision. So let's start with the projection and association fibers. The probably most famous and most important projection fiber system is the optic radiation, and we will start with that one. And you might very well think that you know everything about the optic radiation, but luckily in the very recent years there have been quite some new developments, particularly regarding the use of diffusion imaging, which helped to delineate a bit more than was known previously of the optic radiation. But let's start with the classic anatomical studies. And you see here a delineation on the ventral aspect of the brain. So here's the frontal lobe, here would be the occipital lobe. And you see nicely delineated here this very large fiber bundle based on a Klingler dissection method. Klingler dissection meaning that you freeze the brain and warm it up and freeze again, warm it up again. And this freezing and warming results in a break of the brain along the fibers, because of there is less resistance compared to across the fibers. So if you then try to break up the brain, it will split, particularly along the long-range fiber tracts. And this was used several times, and in this case to delineate and to map the optic radiation. And you see nicely where it starts, around here in the middle of the brain. This is the brain stem. And here would be the lateral geniculate nucleus, which is the beginning of the optic radiation, and then the optic radiation moves all the way through the temporal into the occipital cortex and ends up here in the occipital lobe. And the two arrows point to a particular part of the optic radiation, which would be the so-called Meyer's loop. And Meyer's loop is important because it runs all the way deep into the temporal lobe. All the other parts of the optic radiation are closely related to the lateral ventricle, so in the middle part of the brain. But this Meyer's loop runs all the way to the outer and ventral aspects of the temporal lobe. And we will see in a minute why this might be important to know regarding its functions. But just to show you that this classic technique was actually replaced, so to say, by new diffusion imaging techniques. You can nicely map the optic radiation with diffusion imaging. Here in red, the lateral geniculate nucleus again, and then you see also the Meyer's loop running through the deepest part of the temporal cortex and then all the way back to the occipital lobe. And very recently, a study could show that the optic radiation is not a complete mixture of fibers running from the lateral geniculate nucleus to the very back of the brain, but it can be clearly separated into different parts. And one differentiation they found was that the dorsal aspect and the ventral aspect, here in orange and blue, are clearly separated from each other. So the fibers and the information from the dorsal part of the visual system, and which terminate then in the dorsal aspect of the occipital lobe, here in this case primarily in primary visual cortex V1, would be here on the dorsal part, so we look from the top, and here in blue the ventral aspects, which contain the ventral information from the ventral visual field, which would run here on the bottom part. And again, you see Meyer's loop. Meyer's loop contains mostly ventral fibers. And we will see in a minute why this is the case. Meyer's loop does contain a particular aspect of information from the visual field. Not everything, but a particular aspect. But another differentiation they could establish, yeah, now it was a bit too far. They could also show that not only the optical radiation contains fibers which run to area V1, but also that there are segments which connect the lateral geniculate nucleus with parts of area V2 and also with area V3. And this is not new. It was known before that the optical radiation does connect the lateral geniculate nucleus with areas V1 and beyond. But nevertheless, it was for the first time shown that this could be mapped based on the fibers, the real fibers that run through the optic radiation. So you see there are different segments which run into different parts of the occipital lobe. All right, so this picture you have seen a couple of times today. This is a depiction of the visual system, and you see here the visual field and the eyes, and then the tracts which run from the eyes into the brain. And let's keep a focus on where we were before. So here would be again the lateral geniculate nuclei of both sides, and then the optic radiation coming out of it. And let's imagine you see something like these visual scenes here. For example, you drive by a bicycle through the city, or you read a newspaper, and now something happens in your brain and some of your fibers get destroyed. What will happen to these scenes, and what are you going not to see anymore? For example, if you have, unfortunately, for example, a tumor within the temporal lobe, and this is what I mentioned before, again, the Meyer's loop, which part of the visual system will be destroyed? The problem is, the Meyer's loop does contain the information from the outer visual quadrant of the visual field. So in this case, we are here on the left side of the hemisphere, or the left hemisphere, the left side of the brain. And as you see by the colors, the blue fibers connecting the different parts of the visual field. In this case, it would be the right part of the visual field, which is contained within this Meyer's loop, and particularly here, the upper part of the right visual field. So what will happen is something like this. You will see only the remaining parts, and you will miss what is going on in the right upper visual field, and it's called something like superior quadrant anopia, or in a more common framing, pie in the sky deficiency, because it looks like a pie which has been cut out of your visual field. And you would probably won't see what is going on the street anymore. And this is due to the retinotopy of the fibers. We heard about retinotopy in the cortex, but of course the cortex can only be retinotopically organized if the fibers are retinotopically organized as well. So here the Meyer's loop contains the information which comes from the outer quadrant of the visual field. And the second example would be if you have some stroke or whatever kind of destruction within the whole optic radiation of one side, in this case again the left hemisphere, you would have a deficiency within the complete right visual field, and this would be a homonymous hemianopia, in this case of the right side, and you would not be able to read the right part of the newspaper anymore. So you see, already on the projection fiber level, you do have functional correlates with what is going on in the fibers which run from the eyes into the occipital cortex. So let's move on to the association fibers. So if the information is within the occipital cortex and has been processed within the different cortical areas, how does the information move on into different other areas, not only within the visual system, but also beyond to other higher association areas? And I will show you first two major fiber bundles. The first one, which I want to show you, is the so-called inferior longitudinal fasciculus, here depicted from the lateral aspect and here from the ventral aspect. And you see where it runs from the occipital cortex on the ventral part into the temporal lobe. And here you see how it ends nicely within almost in the temporal pole. And the second one, yep. The second one would be the inferior fronto-occipital fasciculus, very close to the one I mentioned before, to the inferior longitudinal fasciculus, but a bit more medial, as you can see here on the ventral aspect, and it runs from the occipital lobe all the way to the frontal lobe. So we have two major large fiber bundles which are close together, but as you can see here, based again on diffusion imaging, they can be separated. So the question now is, what do these fiber bundles do? You see again here the case in the human brain, and as has been shown recently, it is quite different from the situation in the macaque brain. In the macaque, you only have a very short fiber bundle which runs from the temporo-occipital junction to the frontal lobe, but here in the human, it's all the way from the occipital to the frontal lobe. And this nicely fits to another evolutionary aspect which has been shown by Carl Tillis, that if you map an ape, the brain of an ape, in this case of a bonobo ape, to the situation of the human brain, you see the most developing areas or parts of the cortex marked here in red. And as you can see here from the lateral view and here from the ventral view, particularly those parts which have been connected by this inferior fronto-occipital fasciculus, these are the ones which are best or mostly developed from apes to humans. So maybe this might hint that this inferior fronto-occipital fasciculus, which could not been shown in macaques and other monkeys, might have been developed even further in humans. And how do we make use of this? Why is it necessary to know about these particular fiber pathways? You see here two examples of two patients which suffer from gliomas within the occipital cortex. And as you might imagine, here, particularly in this white matter fiber bundles, there might be those inferior longitudinal fasciculus and inferior fronto-occipital fasciculus. And by intraoperative electrical cortical stimulation, you can nicely delineate those fiber bundles actually, because the subjects, they will suffer from things like alexia, agraphia, or different kinds of aphasic symptoms. And after removal, these are the very same patients after operation and after surgery, after removal of most parts at least of the gliomas, the patients indeed suffered, unfortunately, from alexia and agraphia directly after surgery. After a few months of recovery, they got better because the inferior longitudinal fasciculus has been removed on this side, but luckily the transcallosal fibers, which we will see in a few minutes, are preserved. So probably that the alexia and agraphia got better after a few months was due to the remaining transcallosal fibers which helped to improve these symptoms. But unfortunately, particularly this patient here, had still a lot of paraphasias and anomia due to this large extent of the tumor within this inferior fronto-occipital fasciculus, which would run in this direction. So you see, these two fiber bundles are indeed very informative of functional anatomy in this case, and of particular functions which are associated with them regarding the visual system. And the question is, are there only these inferior bundles? No, there are also superior bundles. And the superior bundles you can see here is this one in green. And very recently this has been described. And the blue one, again, would be the inferior fronto-occipital fasciculus. The green one was a bit controversially discussed, so to say. The colleagues here suggested there might be a new fiber bundle which has not been described before, or maybe in some old anatomy studies, but nevertheless nobody knew about it anymore. But there have been other studies, and they showed a very similar direction and course of such a superior fiber bundle. And this one is very well known. It's the so-called superior longitudinal fasciculus, which has three parts, as you can see here in the different colors. And actually this controversy resulted in the question, is this the very same fiber bundle? Is this the superior longitudinal fasciculus here in green? Or is it something different corresponding to the inferior one I showed you before? Is this the superior fronto-occipital fasciculus? And luckily, the colleagues came to the conclusion that no, there is no superior fronto-occipital fasciculus. This must be the very same one, so there is only a superior longitudinal fasciculus which connects the parietal cortex with the frontal cortex and has some extension into the higher extrastriate visual areas. And what you can see here on this coronal slice through this very last section indicated here at -48, you see that these three parts of the superior longitudinal fasciculus are clearly differentiated from each other. This would be the same actually if you move a bit more caudal to the occipital lobe. And what has been shown by the colleagues here in this 2011 study is that particularly the SLF2, so the second part of the superior longitudinal fasciculus, is associated with visual spatial processing, for example. So a very important fiber bundle for parts of the visual processing and the visual system. Let's now come to the intralobar association fiber. So what's happening within the occipital cortex? So far we only looked at the long-range connections to other lobes. And this is a very old depiction from the end of the 19th century, where different aspects, different fiber bundles running all the way from the dorsal to the ventral aspects and within different parts of the gyra and surrounding the sulci travel through the occipital cortex or through the occipital lobe, connecting different parts of the cortex. And the question now is this real? Do they exist? Because as already mentioned in the talk before, some of those fibers have really been forgotten a long time. And a recent study tried to map these labels on macro dissections and on myelin maps, and it's really hard to see anything, to be honest. Not only based on the beamer, the problem is not due to the beamer, it's really difficult to see anything there. But luckily, there are new techniques, diffusion imaging for example, and as I will show in a minute, the polarized light imaging microscopy, which hopefully helps us to delineate even more of these bundles. And nicely, Kevin Weiner already mentioned this bundle, the vertical occipital fasciculus, which terminates within the fusiform gyrus, and I will just briefly show you where it is located. This is the one delineated here already at the end of the 19th century, which runs all the way from the dorsal to the most ventral aspects, which would here be the fusiform gyrus. And in this study that has already been mentioned by Kevin, you see a very nice depiction again of a dissection, and here in this very hind part you see a fiber bundle running from caudal to rostral, and you see another one running from dorsal to ventral. And this dorsal to ventral fiber bundle would be exactly this vertical occipital fasciculus, while this caudal to rostral one would be again the inferior longitudinal fasciculus. So it can be delineated. And what the colleagues also showed is that it can be delineated based not only in this macro dissection, but also based on classic myeloarchitectonic studies. For example, here, delineated in this very occipital coronal section, you see this fiber path running from dorsal to ventral, and is clearly delineable from the more medial aspects of the optic radiation in the inferior longitudinal fasciculus. And the same is true for in vivo studies using T1 contrasts, where again an occipital lobe coronal section has been taken, and you see here the very high T1 contrast, again as compared to the medially adjacent other fiber tracts. So you can delineate the vertical occipital fasciculus even in vivo imaging. And as you saw already on this very old slide, there is not only the vertical occipital fasciculus, but there are still others, and a lot of other fibers. And based on the new technique which was developed in Jülich a few years ago, the 3D polarized light imaging, it is possible to delineate even more fiber bundles. And this is an example of a coronal section through a vervet monkey brain. And you can see here by the colors different directions of fibers crossing all the way through the occipital lobe. And here in black, you see the stratum sagittale, which contains the optic radiation, and you do see a lot of, for example, U fibers surrounding the death of the sulci, here in this case, and you see, again, nice bundles running from the dorsal to the most ventral aspects, which might correspond to the vertical occipital fasciculus, for example. And as we zoom in, for example, into this region here of the ventral visual system, ventral visual cortex, you see a part of area V3V and a part of area V4, and you nicely see how here the U-shaped fibers run into the cortex and join the radial fibers within the cortex, and you can even see that there are within the cortex these horizontal fibers which tend to have the same direction of the U fibers which are within the white matter. So a lot more to see and a lot more to study if we use these nice images of 3D polarized light imaging. And as the last aspect, we have the transcallosal fibers. And the transcallosal fibers, as I already mentioned, is very important to have the communication between the two sides of our visual field, so for binocular vision. And it has been shown already 10 years ago how very rich and nicely developed these transcallosal fibers are within the human cortex, and you can see here in blue these transcallosal fibers which run through the corpus callosum, here depicted in this dark green. And this particular part is the very caudal aspect of the corpus callosum, the splenium. And all the fibers which connect areas of the visual cortex of both sides run through the splenium of the corpus callosum. And they could also show that these transcallosal fibers connect different parts of the visual system, not only area V1, here in light blue, but also parts of the dorsal and the ventral visual areas. But it was also shown that within V1, the fibers do have some kind of topography. For example, that we have dorsal and ventral aspects, which are separated based on the fiber topography, and again, central and peripheral aspects. But this is only one aspect of what is going on within the transcallosal fiber systems. Another one was proposed already 25 years ago and was based on numerous studies in macaques, where it has been shown that the transcallosal fibers might have some preferential target sites within the cortex. They do not spread diversely throughout the whole visual cortex, but they do have some preferential target sites. And we already heard that there is something called vertical or horizontal meridian within the visual field, and that this is also represented within the cortex. And you can see here in this occipital lobe depiction, here would be the occipital lobe, here would be temporal cortex, fusiform gyrus, and lingual gyrus, and some of the visual areas labeled. And which is highlighted here with these small black dots, these would be the endings of the transcallosal fibers. And as you can see here, for example, at the transition between V1 and V2, you do have a very dense delineation of these transcallosal fibers. And this would correspond to the vertical meridian then. So the transcallosal fibers tend to exactly map the part of the visual system where both sides, left and right visual field, meet at the vertical meridian. And this is not only the case from V1 to V2, but also from the transition to other higher visual areas, although it is not as focused as between V1 and V2. And in a recent study, we could show that this is particularly also the case in humans. This was the first hint, but the authors were not sure if this is really true in humans, because they could corroborate some of the transcallosal fibers, others they couldn't. But in a recent diffusion imaging study, we were able to show that particularly those fibers which run around the lateral ventricle via the tapetum and the forceps major indeed end at particular target sites within the cortex. And here in different colors you see the different visual areas, and here in yellow to orange you see the fibers coming into the cortex. And you see here, for example, at the transition from area V1 to area V2, we do have these endings of the transcallosal fibers. And the same is true for higher visual areas. For example, here you can see nicely the transition where the transcallosal fiber runs at the outer boundary of area V4, for example, or in this case of two of the areas of the fusiform gyrus, FG1 and FG2. Of course, this is not black versus white. there are other fibers which might run within these areas or into these areas, but the major parts have these preferential target sites. And the border, particularly between area V1 and V2, where these transcallosal fibers terminate, is a very special aspect, a very special border. And this has been shown already 50 years ago and has later been confirmed that we do have some kind of transition. So the border between area V1 and V2 is not one clear border, but there is something going on at this particular border. So you see here Gennari stripe again, which has been mentioned before, but you can also see that there is some thickening, some darkening of the so-called inner Baillargeau stripe. And on the other side, when it comes to area V2, you see that there is much more myelination in the upper supragranular layers than is, for example, here or here. And the question is, what is that? It has been termed, in German it would be Grenzbüschel and Randsaum, in English border tuft and fringe areas. So there are some kind of transition areas between V1 and V2, which would be the border would be here. But this border tuft, particularly this border tuft, is the interesting one because this one is the one which contains the transcallosal fibers. They end particularly here. And as we could show in our study as well, based on polarized light imaging, this border tuft is particularly complex in humans. And you again see here area V1 with a Gennari stripe and area V2. And when it comes to the border region, you see that there is some complex fiber organization within this region. You see here a long bundle running from the deep white matter radially into the cortex, and you see here some fading of the Gennari stripe, and this is going on here in the border tuft region. And this would be then again the border tuft on the side of area V2 and the fringe area on the side of area V1. And if we enlarge it a little bit further, we can even see more. You see here labeled the layers of the cortex. You see that this border tuft is even more complex than we thought before, which would be that we do have a part which is directly close to area V1, which has very long radial fibers running into the upper layers, while we have a second part which is shorter and only connects to the deeper supragranular and particularly the infragranular layers. And as a last slide, I would like to show you again a slide of the vervet monkey brain, which nicely shows area V1 here with a Gennari stripe. And here you can better than in the human brain, see how the transcallosal fibers end within this particular border region. And you see here, coming all the way through, the forceps major, which ends particularly here within this region. Here's area V1, which stops here, and here would be area V2. And if we increase and zoom in into this particular region, we see that here the transcallosal fibers directly run into this so-called border tuft region. So we do have a high complexity, and if you even look closer, you see that there are a lot of very small fiber bundles running from this border tuft region into the neighboring areas of V2 and V1. So maybe a long-range connection at one particular location, and then the local connections via horizontal fibers. Thank you very much.
Thank you, Svenja. Any questions? Yeah.
So if I understand correctly, I think that your study shows that your diffusion tract in humans can clearly show some selective projection toward the B1, B2 border or some area boundary border, which is the vertical meridian. So I guess the previous study doesn't really successful about that. So could you explain a little more detail about what the technical detail, what the resolution data, what type of tractography you use? So why do you think this is successful and it's not successful in the previous study?
Yeah, so you heard a lot this morning about diffusion imaging and the different techniques that are possible. We used a quite high resolution, angular resolution diffusion imaging, Hardy technique, with 120 directions and constrained spherical deconvolution to reconstruct the data, so to have as much information as possible about the crossings. So luckily we were able to show these nice fiber bundles in the end.
Okay, so we reached the break. And we will be back precisely at 3 o'clock for the last section. Thank you.